Thank you very much for coming back to our channel. Today we are going to be looking at DNA replication. DNA replication. So what is DNA replication? How does it occur? Why does it occur? Where does it occur? So that's some of the questions we ask on DNA replication. What is DNA replication? To replicate, it means that it is doubling, to double. So like making a copy. So that is the meaning of replicate. But the old and the new must be exactly the same to bring about the meaning of replication. So what is DNA replication? Is the process by which DNA makes exact copy of itself. So when you are writing a definition and you don't talk about DNA making exact copy, if the word exact is not there, then it means that you won't be able to get a tick. So it must have the meaning of, it must have the word exact. So that's why you say that the old DNA molecule must be exactly the same as the new DNA molecule. Then you're saying that when. Now the question is when. When does it occur? When does, when in the cell cycle it takes place? When does it occur basically? Or it occurs during interphase. Yes, you can say during interphase or before cell division, because interphase occurs before the cell divides. Therefore, it is here you have generalized. If you say that cell division, you have generalized. Then interphase, you are just specific to when exactly does it occur, because we have different phases, but the first phase is interphase. So DNA replication occurs in interphase. So where? Now another question is about where. Where does it take place, or where does it occur? Then you are saying that it occurs in the nucleus. So it means that DNA replication occurs in the nucleus. When you're talking about DNA replication in this case, we're talking about DNA replication in eukaryotic cells. We're not talking about DNA replication in eukaryotic cells or in prokaryotic cells, because these don't have nucleus. Then why does it occur? Why should we have DNA replication? Sometimes we ask the significance of DNA replication. What is the significance of DNA replication? That is the same as asking you, why does it occur, or why does it take place? It takes place to double. You want to see the word double? Yes, to double. What is it doubling? The chromosome number. Don't say to increase the chromosome number. Yes, it is correct. But when you are mentioning, we want to see the word doubling, to double the chromosome number, or to double the genetic material, so that now each cell can obtain an exact number of chromosomes. So why does it occur? The answer we need is to double the chromosome number. So if you had 23, if you were haploid, then when you double, then you become diploid, so that now the cell divides. If it is case of meiosis, divides, then goes back to haploid, so that you maintain that chromosome number from generation to generation. Yes. So another one is results in the information of identical daughter cells during mitosis. Remember, cells undergoing mitosis are diploid. If they are diploid, it's supposed to double. The genetic material is supposed to double. When doubles, the cell must divide. Once it divides, then it means that each cell must have the same number of chromosomes. So it will be identical to the original cells. So it means that if I have 23 pairs in one cell, then it doubles, it's going to become 46 pairs. So when it divides by mitosis, it's going to go back to 23. So that is the meaning of to bring about to identical daughter cells during mitosis. So in meiosis, it is always haploid. Then when it duplicates, then it becomes diploid, whereby it is unreplicated. Yes, it is single-stranded. Sometimes call it unreplicated. But after replication occurs, then it becomes double-stranded chromosomes. So it becomes double-stranded chromosomes. We will see this when you go to meiosis. So in exam, when they ask you what is the significance of DNA replication, they brought this question in 2020 November, December still. But in most cases, students, when you ask such questions, they always find a challenge to answer this question. Then, how does it occur? In other words, the process of DNA replication. How does it occur? How does DNA replication occur, or the process of DNA replication? So you have to first be with an imagination in the head so that you can describe the process of DNA replication. You need to first know the imagination of the structure of DNA. Then you have to follow the diagrammatic representations, and then you form the two new DNA which are genetically identical. So using that diagrammatic representation, from there, then you can write the points which are needed. All right. DNA is, we say that is a double helix. Yes, it's a double helix. It means that is two helical, first helical, and then the second helical. So it has two strands. So since we want to carry out DNA replication, this helical must straighten. It must form two strands. Yes. So it must try to break. When it uncoils, it forms this. But I'll show you how do we write in exam. So it forms two strands. Yes, two strands. After forming two strands, now these bonds are supposed to break. So you can say it unzip, or you can say weak hydrogen bonds break. So you can use any of the two because they mean the same. So that is unwinding. It starts to form these two. So now it means that you have two strands which are open. Yes, it was like this. Then because it was coiled like this, so it uncoils, so it forms these two strands. When these two strands, if you bring it here, yes, it opens. When the weak hydrogen bonds between these bases break, then it exposes these two strands. Then if these two strands, when they uncoil like this, then the free nucleotides of DNA from the nucleoplasm will come and arrange. When they arrange, they are forming now another strand on it. This strand must be complementary to this strand. So I have the new strand and I have original strand. Yes, so it means that what is on this DNA which is forming must be exactly to what is this DNA which is also formed, must be exactly the same to this previous DNA. So basically I have three kinds of DNA here, but one is splitting to form two. So this is the which is original. So it means that it has this original strand. Still, this is original. It has this original strand, and then it has this new strand. So at the end, I'll have two new DNA. So I have two new DNA. So it's gonna form like this, whereby it has the original, the original strand here. It has the original strand here, and then it has the new strand. So then it means that you form two new DNA. How do you write in exam when you're asked to describe the process of DNA replication? DNA double helix. So you start by writing the DNA double helix. Yes, it unwinds the weak hydrogen bond. When it unwinds, it forms two strands. So now the weak hydrogen bond break. So I say that you can say weak hydrogen bond break, or you can say unzip. So when you mention one of that, you get a tick. To form now two strands, it forms two strands. Yes, because I want to form two new DNA, then each strand will act as a template for formation of a new strand. So template is like a mirror. Yes, so the old strand is acting as the object, and then the mirror you form another one which is inverse you are seeing. So now this old strand will act as a template for formation of a new strand. Even this one will act as a template formation of the new strand. What do they use to form the new strands? They use the free floating nucleotides of DNA from the nucleoplasm. So it forms two new DNA which are genetically identical, each with one original strand and then one new strand. And then this process is controlled by enzyme. So that's basically what you are supposed to explain when you are in exam. So let's look at it. When we are writing, each different color means a marking point. So you should not forget about it. DNA double helix, we give you a tick, unwind. Yes. Weak hydrogen bonds, they break. Yes. Weak hydrogen bond break, we give you a tick. Or DNA double helix, unwind and then unzips. Then you also give a tick here and give a tick here. So any of that, you will get a tick. To form two separate strands, you will need to see the word two, and these two strands must be separate. So you say to form two separate strands or to form two single strands. So you get a tick for that. And then you're saying that each acting as a template. What is each? Each strand you have mentioned here is acting as a template. If you don't say each, you say two strands. The two strands formed are acting as a template. We want to see that word, either each or the two strands. We must see that. If you don't mention that, then you lose a mark for the template. Using, what do they use? Using free, want to see the word free, floating nucleotides, free floating nucleotides from the nucleoplasm. So when they use free floating nucleotides from the nucleoplasm, what happens? They form what called a complementary DNA strand is formed. As I showed you that it has the new strand. So the new strand becomes the complementary strand to the original one. So it forms the complementary strand whereby adenine pairs with thymine and then guanine pairs with cytosine. The two new DNA molecules are formed whereby these two new DNA are genetically identical. So it forms two new DNA, and each DNA is identical to the other. So basically, the two new DNA are exactly the same. That's why I say that DNA replication, we want to see the word to form exact copy, exact. So that if they are genetically identical in terms of genes, then it means that it won't affect the physical appearance of an individual. So the person won't keep on changing and changing and changing. So it will remain the same. That's why it is not affecting the physical appearance. Then you're saying that with one original strand and one new strand. So the DNA, the two genetically identical DNA formed, each is having one original strand and then each is having one new strand, as I explained before. This process is controlled by enzyme. So basically, that's how you are supposed to write. Here we give you a tick, 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 give you a tick, a tick, a tick, a tick, a tick, and then a tick. Most sometimes we also give you a tick here, but sometimes we don't give, depending on the marking guideline. So that is basically the process of DNA replication, and that's how we are going to ask you. What about DNA profiling?